क्रैनियो को फिल्टर करते हैं
दिव्या
मला पण दिली की कॉल नव्हता कॉल मला पण नाही दिला नव्हता काळजी काय असेल काय
Amal sir, good afternoon.
Good afternoon. How are you?
No, sir. I'm fine, sir. Thank you. How are you, sir?
Yeah, I am also fine.
Yeah.
So when will we starting?
Yes, sir. Within a few minutes, we will start, sir. 2-3
Oh,
minutes. Yes, sir.
Dr. Vaidya, should we start? Dr. Vaidya?
Good afternoon, ma'am.
Good afternoon.
Yes, madam, we can
Guru
start.
करायचं काय?
good afternoon, ma'am. Good afternoon, sir.
Good afternoon, Dr. Suryawanshi.
Amal, कैसा है?
How are you?
अरे
Yeah, I
हाँ,
am fine,
मैंने
sir.
वो-
Good. Nice to see you after a long time.
Yeah, it's really long time.
Shailesh?
बोला सर, चालू करू.
Oh, yes.
चालू करू का?
Yes, madam, we can start.
Ma'am.
Okay. So good afternoon to all the participants and the organizers for having made me the chairman today. I don't know how much justice I'll do, but I would like to introduce Dr. Amal Suryawanshi, with whom we have, at least I have been associated for many years, and he has also been helping us throughout with our research, especially my PhD student, which I'll never forget. His timely intervention was of utmost use to us. So before we go into all the nitty-gritties, I'll introduce Dr. Amal Suryawanshi as an eminent scientist in the field of clinical proteomics. He obtained his master's degree. He obtained his master's degree in animal genetics and breeding from Mumbai Veterinary College and PhD from NIRH, Parel, Mumbai. His major research focus is in disease proteome mapping and biomarker discovery, molecular protein markers in the pathogenesis of several diseases such as rabies, chikungunya, Japanese encephalitis, dengue, etc. He has also organized several symposia and workshops at the national and international level at his institute. He has several peer-reviewed publications to his credit in national and international journals, and he is a life member of several societies, including the esteemed Human Proteome Organization. So with this small introduction, I would like to introduce his topic today. Dr. Amal Suryawanshi is going to speak on Introduction to Advanced Quantitative Proteomics for Understanding of Biology and Diseases. I think, sir, it's a very relevant topic today. And since we have worked on rabies with you, I think you can enlighten us on this topic. Please, sir, please continue.
Yeah, thank you, madam. So first of all, I would like to thank organizers for giving me opportunity to talk in this forum, especially Dr. Jende, Dr. Vaskar, for giving this opportunity to me. So today I will be talking about the proteomics. So basically I will have the two talks back to back. In first talk, I'll try to cover the introduction part where I'll talk about the mass spectrometry based proteomics. What are the basics involved in that, how protein can be identified. And in the second talk, I'll be more talking about the projects, what going on in my laboratory, how we are utilizing these proteomics approaches for the understanding the diseases and the biology. So let me share my slide first. Is this visible now?
Yes, sir. It's visible.
Sir, please put on the slide mode, sir.
Yeah, it is already there. Now it is okay?
Yes, sir, we can all see. Yes, please continue.
Okay. Yeah. So as I told that I will be talking about the introduction to mass spectrometry based proteomics, especially the advanced quantitative proteomics. So first slide shows my institute. I belongs to Institute of Life Sciences, which is located at Bhubaneswar, which is the state is the Odisha, which is the eastern part of the country. So you guys can see this picture. Is there any question? Okay, you guys can see this picture where you are seeing the two different forms of the butterfly. One is the caterpillar, another one is the actually full form of the butterfly. So here if we try to see what is the difference between these two pictures, we'll able to know that the genome of these two, caterpillar as well as the butterfly, is the same. But when it comes to the proteome is completely different, which shows that the different proteome at the different type of point will be present, and which will be most important for the phenotypic changes what you are seeing it. So that is why, as compared to genome, the proteome has a lot of advantages to see these things and understand the biology of the disease. So this is the classical pictures, central dogma. Everybody knows about that. So if you take any cell will contain the nucleus. Nucleus will have the genes, and these get transcribed into the mRNA, and mRNA will undergo with the splicings, which will give the multiple mRNAs, and then it undergoes with the translation, which will form the protein, and then protein further undergoes the post-translation modification, which will give more different form of the protein in that. If you see here, the number of genes is approximately 30,000, but when it comes to the RNA, it much more than the what genes you are seeing in the cell. And then it comes to the peptide, then it is other several billion. So, and actually about body physiology, proteins are very much important as it are more involved in the physiological or the biological processes. This is the another picture which is showing the interaction between the protein and protein. So if you see the human protein, whatever the proteins are there, this is shows that how complex it is. So as I told that human genes are around 22 to 30,000, but when it comes to the protein, it is more than 300,000 proteins or protein variants. And then it become more complex system to understand it. It's not same as the genome to understand it. So we need to give the special attention to see all these things in a different physiological condition. Then only we can able to understand the physiological processes. I'm here showing one cartoon where you are, if you see this cartoon, you will understand that there is a one point which is having a lot of molecules are there, and there are person having the simple tool, which was the before omics era. It is like they were fishing one proteins from the that pond and trying to characterize those proteins. But after proteomics, things had changed drastically, and then the advanced tools which we have got, we can go for the discovery-based approaches, where we are identifying many more proteins at a time, and then filtering it out which are relevant to that, and then characterizing it. So this omics has brought the revolution and the fundamental shift in the strategies from piece-to-piece information to the global analysis. From here, we're getting the piece-to-piece information, like an individual molecule we are characterizing. But when it comes to the omics, now we are trying to identify the global analysis and try to do which are the proteins which are relevant to that particular disease or the biology context. It has also brought the shift in the hypothesis-driven research to the discovery-based research. So hypothesis means we were first hypothesizing that, okay, this molecule has maybe having this function, and then going for the characterization of that molecule. But in discovery, after proteomics things, we first try to discover what are the molecules involved in that, number of molecules, and then further go into analysis and see what is their role. And so it has shifted whole, because of the omics, there has got the fundamental shift in our strategies also and the research also. So I have put some definition which is broadly used in the literature to make you understand what is the proteins and what is the proteomics. To start with the proteome. Proteome is nothing but the protein complement of a genome. Then it can be also defined as the complete set of protein expressed and modified by a cell, tissue, organism at given time or the particular time. Then the proteomics is nothing but the identification of all the proteins encoded in the human genome. This definition was given by the Human Proteome Organization. In 1994, Mark Wilkins has coined the term proteomics, who defined it as the study of proteins, how they are modified, when and where they are expressed, how they are involved in the metabolic pathways, and how they are interact with one another. If you see this definition, you will understand how complex is to study the proteins or the proteomics thing. Because based on the modification, expression, in their involvement in the multiple things are changing. So you need to study each and everything to understand the whole aspect of the particular pathology or the physiology, to understand how these proteins are working in that particular situation. So again, going back to this central dogma thing, why proteome as compared to the genome, as compared to the transcriptome? So if you see the genome is mostly static. DNA tells what possibly can happen in the system. When it comes to the transcriptome, it is dynamic as compared to the genome, but RNA tells us what probably can happen in the system. But when it comes to the proteome, it is too much dynamic, and it actually tells us what actually happens into the system. Obviously, as I told you that there are variability factors are there, like a genomic variation, alternate splicing, protein cleavages, modification. All these things comes into the picture when it comes to the protein or proteome study. So why analyze the proteome over the genome? So to summarize this, the DNA sequences alone does not reveal the biological function. One gene can code for more than one protein. So because of the gene rearrangement, RNA splicing, it may go for the more than one protein when the gene is only one. Then it comes to the, unlike the genome, the proteome is not fixed. So genome, if you study in any cell, it will be the same. But when it goes to the proteome, it varies from tissue to tissue, varies between the different cell types. According to the developmental of stages, it changes. And again, the environmental factor like diseases and other things, if come into the picture, it shows the variation in the expression of level of this proteome. So when it compared with the transcriptome study, it has very poor correlation between the mRNA and protein expression level. Then each transcript can give rise to several number of protein isoforms via the post-translation modification. Then the mRNA expression not indicative of any active cellular protein. So these are the limitation factor comes in terms of the transcriptome when we compare with the proteome. So then the next question comes, how do I identify the protein? What are the different methods available to identify the protein? If we see this, then basically protein identification uses the combined techniques like bioanalytics and bioinformatics for the identification and characterization of any protein. So if you see the literature, the present available methods is the Edmund degradation sequencing, that is the N-terminal sequencing, where people are using this for the actual protein sequencing. But the limitation is that you cannot go more than 20 amino acids, and it is only on the N-terminal side. If N-terminal is blocked, then you cannot get that sequence also. As many proteins are more than 20 amino acids or much more than 20 amino acids. So this method is not useful to identify or quantify the proteins in terms of the understanding the physiology or the disease system. So the next things comes into the picture is the mass spectrometry. So mass spectrometry can do the peptide fingerprints and the fragmentation analysis. So I'll come in detail of this. What is the peptide fingerprint? What is the fragmentation? In my future slide, I'll describe that thing. But and then for all these things, definitely by databases, bioinformatics, algorithm, these are the things which can be used to identify the proteins. So if you see, what are the components of the proteomics? Basically, people say that there are three components of the proteomics. One is the protein separation, where we need to separate the protein first, reduce the complexity. Then the second component is the mass spectrometry. You then load the sample on the mass spectrometer, identify what are the proteins present in that, and once you get the data, then analyze this data on the proteoinformatics analysis or the data analysis or the interpretation. But here I have added two more components, which is also the most important. When it comes to the protein separation, prior to that, the sample preparation is very crucial factor. From what tissue, what cells you are extracting the protein, what are the methods you are using it. If your sample is complex, what are the things you are going to use to reduce this complexity are the much more important when you are using this technology. There are certain reagents which is not compatible with the mass spectrometry. That also we need to think it when we are planning for the sample preparation. So this is the one of the thing. Then the another thing is the after this proteoinformatics analysis, you will get the many more number of proteins, but not all proteins are directly relevant for that particular. It may be the indirect response. It may be the response due to some other factors. So we need to validate or verify this data once you land up with those molecules, and then show it that yes, this is the proteomics data which we have got it, and now it is validating with a more number of samples and all the different techniques, so that we can pinpointly say that these are the important molecules in that particular term. So there are the classification based on the gel-based approaches and non-gel-based approaches. So I'll go in detail of that. So here you are seeing this the short classification based on the different approaches. So when you take the quantitative proteomics approaches, there are gel-based approaches, and then now gel-free or non-gel-based approaches are present. So earlier days when this non-gel-based approaches was not there, it was actually started with the gel-based approaches to separate the protein. In that also, there you can see that this can be further sub-classified as a labeling approach and the label-free approach. So this label-free word terminology has come just now, but earlier days it was like started with the gel-based approach. You must be aware that if you get a protein sample, you need to first separate these proteins. So one of the method of the separation is the 1D electrophoresis, that is the SDS phase. So you get the mixture of protein, you run it on the gel, and then you get the separate band on the gel. And once you get this band, then you can go for the identification of this, what is the protein present in that band. And then, so these were the traditional methods. When proteomics come into the picture, many people have started doing the 1D, then cutting the band, then identifying it, and then seeing it that whether these molecules is relevant for that particular system, what they are looking for. Eventually, slowly, people understand that 1D electrophoresis is not sufficient, because 1D band contains is not a single protein, it contains many more proteins. So if your protein is not abundant, you may not able to identify in that band easily. As many methods what mass spectrometry uses give the most abundant protein first. Okay, most likely to identify the most abundant protein as compared to the low abundant. So people thought it will be better to separate this protein sample in the two different ways. So then 2D electrophoresis come into the picture, and then people started using the separation with the 2 dimension. So eventually they understand that there are some difficulties with the 2Ds, and then things comes into the picture, which is the labeling approach. So to avoid the reproducibility issues, then the sample 2D diet, that is 2-dimension differential gel electrophoresis, has come into the picture, and people started labeling their samples, then running into the gel, and then separating it on the 2D to get it. So some of this technique I'll be talking details in the future slide also, but I'm just giving the overview first so that you will understand about how they are classified, how they have evolved from time to time. Then it comes when scientists understood that there are again limitation based on the gel-based thing. So people try to think that okay, there should be something different than this. So then non-gel-based or gel-free approaches has come into the picture. So earlier days, first they started with the labeling approaches. There are different types of approaches people use, like ICAT, isotope coded affinity tag, then the stable isotope labeling with the amino acid in cell culture, that is SILAC. SILAC is mostly used in the cell culture system only. So this is having the limitations in that. Then another 2 approaches has come into the picture is the isobaric tagging, relative absolute quantification, that is called ITAG. So this is the most commonly used approach nowadays, because this is you can do the differential proteomics, compare the different type of sample, and then able to identify the proteins which are relevant in that pattern. This tandem mass tag TMT also similarly works like the iTrack only, but this is the different company has generated this one, and iTrack is having the different company. So, but basically the principle of these two techniques are same. When it comes to the label-free approaches, so some people could identify some more approaches like the spectral peak intensity, the spectral counting, then the absolute quantification, then the multiple reaction monitoring MRM, and then the SWAT, sequential window acquisition of all therapeutic mass spectra. When it comes to the SWAT, this is the label-free, gel-free approach, but it is very specific to one vendor and then one instrument right now. You cannot do it with any other instrument. So there's a limitation. If you are having that platform with you, then only you are able to do it. That is the reason it is new thing, but it is taking time to use by the various scientists. Then the MRM is the technique where you need specific standards, and every time you have to run the standard and see where your protein are getting eluted, and then you have to decide the system where you can identify that molecules, and that is the reason it can be used more specifically in the where very few molecules are you wanted to look at. Okay, that is why this is not used for the global identification. This is more like a targeted approach. Recently, the spectral peak intensity, spectral counting has come into the picture, but that will take little more time to get the momentum to use this thing, because as this is having the still having the lot of difficulties to use this one. So coming to the next slide, where I'm showing you the basic proteomics analysis scheme. Okay, so as I was talking to you that once you get the protein mixture, you need to separate it, whether you use the gel-based approach, liquid chromatography-based approach, and then separate, and then you get the individual protein. Then once you get these proteins, then you have to go for the enzymatic digestion. Then once you get digest this sample, you will get the multiple peptides from the protein. Once you get this peptide, then you can load it on the mass spectrometer and then get the peptide masses out of it, and which will give the MS spectra as well as the MSMS spectra. And then you give this for the database search, and then you identify protein. This is the basic workflow or the basic scheme where how mass spec involved in that. So we'll go in slowly how it works basically. So in my future slide, I'll show you how the mass spectrometer is works for the identification. So this slide shows you that there are three basic components of any mass spectrometer. If you see the literature, you will get confused because there are a lot of different terminologies or names are used for the mass spectrometer. But let me tell you, it is very simple thing. There are three basic components of any mass spectrometer. One is the ion source, the another is the mass analyzer, and then the detector. So if you see it, there are different type of ion sources out there. One is the MALDI, that is nothing but the matrix assisted laser absorption ionization. Then another source is the ESI, that is electrospray ionization. Then it comes for the nano ESI, that is nano electrospray ionization. Then the APCI, atmospheric pressure chemical ionization. So these are the different ion sources where the ionization takes place. Then second component is the mass analyzer, where the time of flight is the one of the mass analyzer, then the quadrupole is the another one, ion trap, then the orbital. These are the different types of mass analyzer. And then definitely it will have the detector. So when any mass spectrometer, if you see, it will have the ion source and then different mass analysis. Sometimes it is a one mass analyzer, sometimes it's a more number of mass analyzers present in the one mass spectrometer. So based on that, the name will be defined. So how it works, I have given it in a simple word, is like a sample can be introduced directly via solid probe or in case of mixture by the intermediary of the chromatography device. So either we can use the liquid chromatography or you can directly put it a solid probe and then you can introduce this sample. The once sample is introduced in the source, the sample molecules are subjected to the ionization. So ionization will take place. The ion forms in the source, acquires some kinetic energy and then leaves the source. So once it leaves the source, the analyzer then analyzes the passing ions function to the mass to charge ratio. And then the ion exiting the analyzer assembly then detected by the signal as registered in the detector. So basically, when these molecules get ionized, then it will be passed through the analyzers, and in the end of the analyzer, the signal will be registered, and then time taken for this molecule will tell you that what is the mass to charge ratio for that particular molecule. So there are some examples of the mass spectrometer here. As I have already talked that there are different types of ionization process. That this one the MALDI, this one is the ESI. These are the commonly used ionization methods for the recent mass spectrometer. Then you will see it here is the first is the TOF. So which is having the time of flight, like one analyzer. But when you go here, there are two TOF. One is the TOF 1 and another one is the TOF 2. So you will see that this is called TOF. So if you see the literature, there is a terminology used as the multi-TOF. So when if it is only multi-TOF, so you should understand that okay, this the multi is the ion source, where the top is the one analyzer is there in that machine. But when it comes to the multi-top, one should understand that okay, this is having the two analyzers. I'll come later in my presentation that why two top is required or more analyzer is required for the biological analysis. So this is the way we can identify what are the composition of that mass spectrometer by looking at what they have given into the literature. The another combination is like a triple quadruple. So here we are seeing it that the three quadruples are there. The quadruple 1, quadruple 2, quadruple 3. So this is the another composition. Then when it comes to the here, then quadruple time of flight. So there are two quadruples and the another one is the top. So in this diagram, we are seeing that this is the example of Q-top instrument. Then it comes to the another analyzer is the ion trap. Sometimes instead of the three analyzers, quadruple analyzers, they will use it two quadruples and one ion trap. And like this, some combination can be used in the machines. Or you are seeing into the recent time whatever vendors are selling as a mass spectrometer. The recent one come into the picture is the Orbiter. Orbiter is also one of the analyzer, and it can go in more depth to identify the molecule. So these are the few examples of the mass spectrometer where you are seeing that two analyzers are used. That is the dihybrid. But now commercially one more instrument has come into the market, which is the trihybrid instrument, where there are three analyzers are present. One is the orbit trap, then the coal, and the linear ion trap. So as different instrumentation has different ability to analyze the molecules. Based on that, if you need to cover all kind of application in one mass spectrometer, you need to have multiple kind of analyzer, then you can do it in one mass spectrometer. Otherwise, for different type of application, you need to see with the different technologies, and then you need to select which one I will go for it. So why it is like this? This is the slide for that. This mass spectrometry, these are the four characteristics where this mass spectrometer differ from each other. One is the speed, another is the sensitivity, then the accuracy and the resolution. Some mass spectrometer are the very sensitive, having the high resolution. These are the mass spectrometer which are good for the discovery based application. when it comes to the accuracy, there are some mass spectrometer or some analyzer which are there, which is more accurate but not that much sensitive. That kind of things can be used for the targeted based proteomics approaches. So that when you are looking for only 20 molecules, which you are not interested, then their sensitivity is not the requirement. There is only accuracy is the requirement. In that kind of application, you can use the mass spectrometer which is having the more accuracy. So these all these whatever combination I have shown differ in based on these 4 characteristics. So as I was telling you in basic workflow, that we have to go for the enzymatic digestion. Why enzymatic digestion is required and how it can be done. So specifically, you cannot load the sample directly, protein sample directly on the machine, because it will not give the much more information rather than the molecular masses present in that particular sample. But we are interested to identify the proteins out of it. But mass spectrometer only give you the masses. Just getting the masses will not help you. So what we can do, we need to generate the much more information from same sample and then combine this information and then analyze and then identify it. So one more steps come into the picture, that you take the protein sample mixture and then you digest it. If you are using gel-based approach, then it will come the gel-based enzymatic digestion. If you are using gel-free approaches, then in solution enzymatic digestion. And the most commonly used enzyme is the trypsin. Why it is the trypsin? Because trypsin cleaves specifically to the C-terminal side of the arginine and lysine. Whatever the peptides it will generate, which will be size of 2 to 3 kilodalton. So when you look for the small peptide, you have more chances that you can identify those peptides and then go for the further fragmentation. The frequency of tryptic cleavage guarantee you the most peptide will be of this size. So when you go for the trypsin, it is very specific, and then it will give you the specific sizes. So you don't need to really look at the big chunk of the things, where if you look at the small size also, you can get this able to identify those majority of peptides, and then you can put this information for the identification search. Then these peptide fragments are more predictable manner throughout the length of the peptide. If it is a random, most of the time you will not able to predict which is this peptide. So it should not be random, it should be the predictable manner. So there are other enzymes also, that chymotrypsin, pepsin, arginine C, lysine. So depending on the requirement, people may change the enzyme if there is some problem with the trypsin. So we can change the enzyme based on the need, but most commonly used is the trypsin. So coming back to this ionization process, once you get this sample which is already trypsin digested, so you will have the peptide mixture from the proteins. So what in the multi we do, we take this plate and then we'll spot our sample on the multi plate along with the matrix. Matrix is the substance which help to ionize the sample basically. So we have to mix it. That is why it is called matrix assisted laser desorption ionization method. So when sample is spotted in the plate, you are fighting with the laser. This matrix will acquire the energy from the laser, and then it will transfer on the ions which are there in the as a sample. Okay, and this ionization process will for where they will just transfer this proton ion from matrix to the sample molecule, and these sample molecules will get ionized, and then as it get ionized, it will acquire the kinetic energy and then fly into this mass analyzer. As you are seeing it here, when it starts at starting point of the analyzer, and if it is of different sizes, you will see that when it runs further, the small molecule will reach to detector faster than the higher molecule. So the small one reaches to the detector before the large one, measure the time for the ion and reach to the detector. So time taken to this masses to reach to the detector will be calculated, length of this tube will be calculated, and based on that we will able to identify the what is the mass of this molecule. And mostly when we say mass, it is the mass to charge ratio. It's not a single mass. So get this is the formula. What we get is the mass by charge. Z is the charge. My m by z is equal to 2p square k per L square, where the t is the drip time, the L is the length of this analyzer, M is the mass, K is the kinetic energy of the ion, and the Z is the number of charges present on that. So this is the way you can get the mass of the particular peptide from this spectrometer. So usually when you get to know is the only the masses from the mass spectrometer. I'll come in my future slide that then how we can able to identify the sequence out of it. So this is the schematic presentation of 4700 proteomics analyzer with the top. So as I was telling you that multi-top contains the two analyzers. This is the first top analyzer. This is the second analyzer. Here is the detector. And then in between these two top analyzers, there is a time ion selector. So in this, when you analyze, spot this sample, put the laser, it will get ionized here and then fly in this analyzer. So when it reaches to the detector, the masses will be generated. So you will get, when you plot this spectra, whatever masses you are getting, that is called the peptide mass fingerprint or MS1 spectra. But when it comes to the MSMS, what we do, we select one of the peptide and tell the system that we are only interested in this peptide. So there is the time I am selected. When it gets separated in the first top, then while passing through this time I am selected, window will only open at that time when these masses will pass and then go to the CID chamber, which is the collusion cell basically. And rest of the time this window will be closed, so all these molecules whatever is passing will fall down there. So whatever the desired peptide will come in this collusion chamber, you will put the high voltage, and then based on the high voltage it will get a random fragmentation, and as it is already having the kinetic energy, it will again go to the this detector, and it will detect these fragment masses. Now it is not the peptide masses, it is the fragment of the peptide, which is also called the daughter ions basically. So you can get the MS2 spectra here. So now you are generating the second information from the mass spectrometer. One is the MS1 spectra, second one is the MS2 spectra. Okay, so this is the way this mass spectrometer generates the masses from the biological samples. So here is the latest proteomics 5800 multi-top. As earlier system you have seen, this is the horizontal. That is the old system. But now they have put all these optics in the vertical manners, and then here is the actually the sample plate, and then this is the way it will get one top, second top, and then it will get analyzed. Okay, so this become a this is the compact latest issue, which is much more advantage than that. I have put that picture because that was easy to understand for you guys. So as I have, what is MS and what is MSMS? So MS is the MS1, MSMS is the MS2 basically. So just recap on this, like this is your sample. You go through the different types of ionization process. Your sample peptides will get ionized and then get with the separated. You will get the MS1 spectra here. That is the precursor ions. And then when you go for the selection of one ion and then further fragment it will give the daughter ions basically, and then it will generate the again separate it, and then it will generate the MS2 spectra, and then it will detect it. So Similarly, here we can see this is the sample run on the gel, then band is cut. This is the protein. Which is, further fragmented, and this now because of fragmentation, now peptide has different fragments. You are seeing it when the MS peptide, you will see it like this, like a, that is a peptide fingerprinting. And then when you select one peptide and then further fragmenting, you will get the different MS2 spectra for each of the peptide basically. So this is the way you can get a lot of masses from this mass spectrometer. So why MSMS is important? Because peptide mass fingerprinting are not sufficient to identify protein only in the cases where the sequence of a protein is already in the database. If protein is already database, then only you can rely on this MS1 spectra and then tell that this is your same protein. But more majority time there are more proteins which is of same kind of thing. So it may mislead to the false identification. So that is why nowadays people are not rely only on the MS data. Therefore, the MSMS is also important along with the MS data. So this MSMS can obtain the partial sequence information from each peptide of the fingerprint within the mass spectrometer. Each peptide can be then further broken down into the ionized fragment. That I have told you how it happens basically. The goal is to produce the ladder of the fragments, each differing length by one amino acid, because each amino acid has different molecular weight and sequence of each peptide can be deduced from that. So let me explain it a little more. Okay, so as in MS spectra, you got the fragment mass, which is the 1410 Dalton. Okay, now you got this peptide mass, but based on the peptide mass, you cannot tell that what is this peptide sequence or what is this protein. So then you go for the MSMS spectra. When you go for the MSMS spectra, this is the random fragmentation. So for example, if you see this amino acid sequence here, and then it may get fragmented at after the first amino acid, then you will generate the B ions which is intact in the N terminals and Y ions which are the intact in the C terminals of this this peptide chain basically. So if it is get fragmented after this one amino acid, then you will get the masses of this yes, that is 81.01, and another mass which will be the rest of the peptide sequence. Similarly, if it get fragment with the at the second one, then you may get this one. So like this, if you plot a graph of the masses identifying from this MSMS spectra, you can identify the pair of the Y ion and B ion, which is giving you the actual sequence. How you can identify? You know the precursor mass. What is the mass of this peptide? 1410. If you combine this mass of 81 plus this mass of this 1323, then total of these two BI 1410 Dalton. Okay. Similarly, you need to identify the another pair. Like this, you can identify all the pairs in your data and then plot this one like a consecutive B ions, consecutive Y ions. And then when you plot it, when you get these masses, then if you see the B1 and B2, if you make the B2 minus B1, you get this mass, you minus this one, then you will get the mass of this proline. And this all this amino acid is having the fixed masses, monoisotopic masses that we know it is there in this table. Okay, this is the highlighted here in this one. Here we can see that this is the proline is having 97 Dalton mass. So if you make it this B2 minus B1, you will get this mass of 97 and then you see, okay, 97 is proline, then you can deduce the sequence like this second molecule is proline. Like this, you can identify all these amino acids. Similarly, you can go with the Y2 minus Y1, Y3 minus Y2, like ladder manner, and then confirm it that whether it is the same amino acids. So this way you can identify what is the sequence of that peptide. Yes, manually it is not very easy. So there are softwares to do that thing, but one should understand how it works basically. So this way you can get the actually sequence of the peptide. So then how we can land up with the protein identification? So definitely you need to go for the database search to identify the protein. So the basically this database search works based on the matching thing. So matching, how it is like this, when you get the experimental masses, like you are having the gel, you cut the spot from here, then get the intact protein, then the enzymatic digestion you do, and then you get the peptide fragment. So you will generate the MS spectra. Then again you go for the MSMS spectra, and then you get the all these experimental MS as well as the MSMS information. And then now you are putting this MS as well as MSMS information for the database search. So how this database works? So we should know that what is the source of this sample. Then you take the database of that particular source, whether it is a DNA sequence database or the protein sequence. If it is a DNA sequence database, you definitely you need to translate into the protein, and then only you can search. Or if you now can go for the protein sequence database, then it will be directly searched. Then in this, when you give database, you have to define certain criteria also, what experimentally you are doing it. Okay, so what are the conditions you are applying experimentally? Same condition you tell to the software that yes, we have our sample gone through these different conditions, and then we got this spectra. So what this software will do, that will generate the theoretical peptide masses based on those conditions, and then make a list of the masses. Now you are having the list of the experimental masses, and now you are having the list of the theoretical masses, and then it will be compared like this. Okay, so now you are seeing it here that this is the experimental masses. Here you are seeing the observed masses. Observed masses are the monoisotopic masses. As I have already told that this goes through the ionization process, and if it is a +1 charge, then mass will be increased by +1 for all the molecules. So these are the observed masses. If you minus that ionization charge, then you will get the experimental masses, and then this is the theoretical masses. Now you are giving this one to algorithm to match it with the what are the masses are matching. So now you are having the two information. One is the MS masses, and another is the MSMS masses. So where you see that these MS masses are matching closely to each other. As you are trying to match it, definitely you have to give some window for the error also, that okay, this I'll be considered plus minus 100 ppm or 0.1 Dalton as the same masses, because this is the statistical analysis. You have to see that okay, you won't get the exact accurate mass in the experimentally as well as the thing. So first important thing from any mass spectrometer data, you need to get the as much as accurate mass from that data, and then you use it for the comparative analysis. So then whatever the window we have given, if it falls into that, then it will show the match, and then it will generate some score. So where you are seeing the score, that is the generated through the ion score, which is the MSMS score basically. And then if it is mass is matching theoretical mass, you know the what is the peptide sequence of that. So you can derive the sequence and then get okay, now this is matching to this protein. So as I told you that it should be the accurate masses from the mass spec. So here is the example. If you get the mass like 1529 Dalton, and if you give the error, which is the 1 Dalton, you may land up with the number of proteins identified is the 478. So there are a lot of false positive in that. So you are opening the window for the false positive. So if we get the mass accuracy of the 1 decimal, and then we go for the mass error, which is 0.1 Dalton, definitely your accuracy of identifying the protein will be much more as compared to the earlier case. So now you are seeing it that 164 proteins are getting identified. Still, it is very high number to say that this is my protein, because this is not the single protein. So the moment your accuracy of the mass will increase in the multiple decimal, uh, and then the mass error will be less, then identifying the correct peptide or protein will be more chances will be there. So that is what you should get, the more accurate masses. That is the first thing. And then second thing is that you should get the multiple peptide masses for that particular protein. Like this, in this example, we have got only 1529.73 Dalton mass. And when we search with the against the database, it is showing that 204 proteins are matching with this mass. So obviously, this it is difficult, which is exactly your protein from this is not possible to tell. When it comes that no, I am having the two masses, we are getting it like 1529 as well as 1252. So we'll tell the database search that no, we are getting two. You search where the both masses are getting into the theoretical masses are getting in the protein. Then when it search, it gives you that there are 7 proteins are matching to that. So number of peptides identified from the your sample, if it is more and it is matching to that particular peptide, there will be the more significant outcome basically. So that is what we say, that if we can able to identify more number of peptides from same sample, then your accuracy of the identification of proteins will be much higher than as compared to this. Otherwise, you need to do something else to come to the conclusion that which is your exact proteomics. So as I was talking to, this is the database search basically. So obviously we need to use the some programs, algorithms for the identification of this. So there are a lot of programs people has developed for to analysis this proteomics data. There are some programs work with only with the MS1 spectra. Some programs work with the MS data. Some programs work with the both of the data together and give a combined search. The most commonly search engines are the Mascot and the CPACE. So in case of multi-top data, people more preferring the Mascot as a search engine, while the CPACE will give the most MSMS information, where the LCMS data you can get the more number of that. So people are more prefer the CPACE kind of analysis. But nowadays, if you see that the vendors are having their own algorithm, which they are saying it that more advantageous. So sometimes we go with the machine specific algorithms to identify the protein. Then it comes to the database. So you should know that what database I should use it for search. If your sample coming from human, and if you are using some database which is from some other organism, this protein may be present in that or may not be present in that particular database. If that protein is not there in that database, if you search against that database, you will show that there is no hit from that particular identification. So you will be clueless. You are having the data, but you are not getting it, which is this protein. So right, database searching is the most important. So if you see that there are lot of databases available in the internet, but most commonly used is the SwissProt and Trumbull. So this SwissProt is and the Trumbull is the UniProt knowledge base database, where the SwissProt is well annotated database, and the Trumbull is like all the sequences translation about the nucleotide. So anyone is identifying any protein, new proteins or new peptides or those things, they are putting into the database. It will go into the Trumbull database, and once the curator will curate that one and see that this is having the evidence that these proteins is experimentally they have shown it, then they will annotate it and put it in this SwissProt. So if something you can able to identify in SwissProt, this will having the more kind of characterize the protein, But if it is you are able to get into this, then definitely there will be not much information available on that. So there are, you can work much more on that. Similarly, now earlier it was the NCBI NR, now it is become the NCBI Pro. So NCBI is like a non-annotated database which contains the DNA sequences from the gene banks and also sequences from the PDB, SwissProt, PIR database. But obviously, if your database is big, then your search time will be high. Sometimes it takes hours, sometimes it takes the days, sometimes it takes months. So depending on what you are looking, you have to see. And definitely, if you are land up with some proteins based, with using this database, it is good. If you are not landing up, that means this might be the completely novel protein. So then you need to do some kind of de novo sequencing to identify those proteins. So as I was telling you that when you go for the database search, you give some information how you process your sample. What is the source of this sample? Which database we need to look? What enzyme you have you? Based on that, then it will generate the theoretical masses. Then what kind of miscleavage you are expecting? Then what is the mass error? What you are expecting? What is the peptide charges present in this MS level as well as in the MSMS level? So if you feed this correct information, then only you will chances of landing up with the correct identification will be more, or you will land up with the false identification or the not able to identify it. So similarly, we can also define the modifications. If you are interested to identify post-translation modification, then you can say into the database search, we are looking for this. You also search for it. Obviously, the search time will be more higher as compared to the normal search. So this is the example of the MS spectra as well as the MSMS spectra. The top one is the MS spectra, that is the PMS spectra, where if you see that one particular peptide is selected and the further fragmented and got this MS spectra, which is the daughter ion spectra. Here the fragment mass is this precursor ion mass, that is the 2253.155. So when you get the data, this MSMS spectra, one thing is there that whatever the precursor ion mass, peptide mass is there, which you are using it for fragmentation, no peptide will be bigger than that because you are fragmenting that peptide. So you will get all fragment masses lesser than that. Okay, so you can use these masses and then you will identify the spectra. Usually, majority of people send the sample outside and then they get a result output like this. So how to interpret this result output? Once you get the protein identification data from mascot result, this is just the example to make you to understand how you can interpret this. So here you can see the top score is the 928 and identifying this one protein that called the beta gal. Okay, so how top protein is generated? It is basically the total score. Total scores come from the MS score as well as the MSMS score. So here you are giving it MS data also, MSMS data also, combining it is searching and it is. So then it comes that whether this score is significant or not, whether I should consider this hit as a significant hit or not. Then this comes into the picture. Then here the software also shows that the protein score greater than 74 are the significant. You look at that, what the significant value to scores should be there for that particular search. So it is not a constant, it changes from database to database. How many molecules are present in your samples, whatever. It has lot of parameters are there. So, but the software tells you that, okay, you consider more than 74 is the significant. So now we can tell that this is highly significant. So there, our identification is the significant. But when you get the data like this, like where you are getting the protein score 129, so this is also the significant. But when your peptide or the protein lie in this green zone, this is the non-significant zone. So when you get these peaks into the non-significant, that means it is having the score less than 74. So whatever you are identifying is not significant. So you need to repeat that one and then identify it. But if you get this kind of score, you should be very happy with that, and then try to analyze it further. So as I have already described this slide, that this is the masses generated from the MS as well as the MSMS spectra. So how you can find out which is the MS spectra? So where if you see this iron column, where you are seeing the iron score, that is the MSMS score basically. So this MSMS score combining with the MS score give you the total score of the identification. So if modification is we have given, then if it is identifying some modification, then you can see it here. Okay, this peptide is the modified one, and this mass is the modified mass. So likewise, you can able to identify this protein. Along with that, you also get the coverage of the protein. So this is the complete amino acid sequence of that particular protein. And then you are seeing it that this red are the peptides which are identifying through the experimental masses. So red one are matching to the experimental masses. So it is showing that our 41% data is matching to this amino acid sequence. So when it comes to the coverage, there is no thumb rule for this that okay, this should be this more than 20%, 30% like this. If your sequence is the bigger one, you might not get all the peptides all the time. So you may land up with the less percentage of coverage. But when it is a smaller peptide, there are likely to be identify more number of peptides from that sample and then get the more percentage. But it will give us the idea that what are the peptides which is actually coming from your experimental sample. So this is how we can interpret and analyze the data. So I'll shift the gear from the understanding the mass spectrometry based approaches to the second part of this first talk only, where I'll show you the what are the different classifications which I have already talked to you about these different approaches. I'll just brief about those two approaches which is shown it here. So let's start with the 2D electrophoresis approach. 2D electrophoresis is the two-dimension polyacrylamide gel electrophoresis. So it was first introduced by O'Farrell in 1975. So basically, as name suggests, it is a two type of separation of two-dimensional separation. In the first dimension, we are separating our protein based on the isoelectric pH of the protein by using the isoelectric focusing unit. And then in the second dimension, the protein gets separated based on the molecular weight or the size on the SDS page. So it has unique capability to resolve the complex mixture of proteins. As I told you that reducing complexity is the most important thing to identify more and more number of proteins. That is the why the when 1D was not sufficient, people move into the 2D electrophoresis. So it also allows you to see the whole proteome in a one go. So it gives you the simultaneous analysis of the protein. So again, when you go for the protein isoelectric point, if you see the human protein, basically most of the isoelectric pH of the protein lies between the pH 4 to 9. Okay, so if we use the broad range strip pH strip, which is 3 to 11, which we can able to identify good number of proteins in that. Similar way, if you see the molecular weight distribution of human protein, majority of them are in the range of the 20 to 90. So as it is fall in this range, we can able to then separate these proteins better way. Yes, we may lose some protein, but majority we can able to identify. So this is the classical workflow of the 2D electrophoresis, where IPG immobilized pH gradient strips are used. This is basically the dry gel. Put it on the plastic strip, which is having the different pH gradient on the strip. So it starts from the 3 pH to the 11 pH. So when as this is the dry strip, you need to rehydrate this strip and then along with the sample. So you are applying the sample also along with this rehydration buffer, and then you are putting this in this isoelectric focusing unit where you are putting the high voltage. So due to this high voltage, the proteins will start migrating towards the isoelectric point. and the moment this they reach to the isoelectric point where the this voltage will be 0, and they stop migrating further. So this will get separated in the isoelectric focusing, and then it goes to the second dimension where it will get separated into the molecular weight. Then you get these kind of gel images. So for the each sample, you have to generate the different proteome, different gel, and then get the different proteome and then compare it. So if you see the classical 2D experimental design, so if we are using the comparing two groups, one group is the control sample, another one is the treated or the disease, whatever it is. So if we are taking the triplicate sample, taking in the triplicate, definitely you need to use the 3 gel for the control, 3 gel for the treated one or the another group. And then you have to run the 6 gel as you are running the 6 sample different in the different gel, and then scanning those gels, then using the softwares like this image master, PDQuest, and analyze the data by intra-group and the intergroup comparison. So what you will get it basically is the what are the expression level of those protein in the one sample, another sample, and then if one group to another group, any variation is there, like upregulated protein down, or is there? So you can get those protein spots. So as you are getting in these protein spots, basically this is important to compare different profiles to get the different cell stages, normal versus disease and disease versus. So you can able to identify them. And then once you identify the proteins here, like this is the example, this is the control sample, this is the another state, maybe you can consider disease sample or treated sample or the different state. Then you can check the proteome of this versus this, and then you can see that which are the differentially expressed protein, upregulated and downregulated. And then you can more further focus what is their role in that particular disease or the particular treatment. So then you can cut that spot, then go for the mass spec, then identify what is this protein, and then you can further downstream processing in that. So basically 2D helps you to get the global screening of the complex sample, provide the qualitative, quantitative evidence changes, and the protein expression in the different situation. It also identifies the target for the further investigation or the validation. But as I told you that these gel-based methods have the limitations. So what are those limitations or the critical factors? This is the multi-step procedure. You are running different gels, so there are the chances that you are in variation will be there, and this variation will be lead to the wrong quantification or the identification of the protein spot, and then it will lead to the wrong identification of the protein. So it is not the reliable data based on the quantitative data, and definitely you need the validation. As it was, people started with that, then people has gone to the another method, which is the 2D dyes, that is the differential gel electrophoresis. It is similar to the 2D, but here the sample is labeled with the some side eyes. Okay, these are the fluorescent dyes. So this technique was introduced in 1997 by Minden Group. So how it works basically, the labeling of the two protein sample with the side eyes and then mixing the sample before the application into the 2D gel. The same isoform of a given protein from the each sample will thus migrate to the same position of the gel. So as this is the label, think it will migrate at the same position. Then the relative abundance of each protein in the sample can be obtained by scanning the gel using the excitation emission wavelength unique to that particular dye. So what we are doing is that we are labeling these different samples, then combining it together, and then running in one gel, and then you are scanning these gels with the different wavelength, we using the excitation and emission different wavelength, and then you will get the, if you are going for the Psi2, Psi3 and Psi5, these are the dye which is commonly used. It has different excitation emission. So you will get the three different images which is showing the representation from that particular sample. So that you can able to get the, from one gel you can get the three different profiles, and then you can compare which are the differentially expressed. So why these tie-dye labelings are used? These are the basically specific properties of spectrally labeled dyes, which is dyes flow. Basically, in this site 2, site 3, site 5 are the minimal dyes is used. Then the side dyes has the great sensitivity. So then detecting the as low as 125 picogram of protein and giving the linear response. Then you can further go for the detection using the silver staining also with this. The labeling does not interfere with the mass spectrometry. This is the most important thing. The minimum labeling can be post-tense after the this sample can be the gel can be posted and then able to identify which is the spot which is differentially expressed and then able to go for it. So just give the brief idea about that how this chemistry works. Basically, this is the dye which is having the NHS ester reacting group, which are covalently bond with the epsilon amino group of the lysine protein via the amide linkage. And then once you link with the protein, and then this labeling will be used for that. So if you go for the minimum labeling, then basically you will have the three types of dyes: site 2, site 3, and site 5. In most of the cases, site 2 we use it for the internal standard, so that gel to gel variation, whatever if we are getting, all is gels, we are using the same internal standard, which is the representative of the all the samples, and then normalize the all the values we get it from the each gel against these internal standards or and then get the expression. So quantitation level will be much better as compared to the normal gels. So here we can see that these are having the different excitation and emission waves which are there. So after this emission, this is how I have already talked about the pool internal control, which is the representative of all the sample basically. So when we compare this experimental design with the 2D versus the dyes, as we have already seen that if we are having the 6 samples, you have to run the 6 different days. Where in this, we are labeling sample 1 with the treated one with the site 3, site 5 internal control with the site 2, and then combining these 3 and running it on gel 1. Then second set of sample is combined and then run it on gel 2 sample. As this cell is having the internal control is common, then you can normalize the value and then you can have the gel to gel comparison. So you get this kind of result. So you are having the site 2, site 3 and site 3. It is combined together, then get this kind of profile when you go use the laser scanner and then get the site 2 profile, site 3 profile, site 5 profile, and then you can find it out the expression level of that particular protein spot, and then find it out which are the differentially expressed protein, then code it and then go for the identity. So this is the example where we are showing the control samples, the internal standard word profile, as well as the another sample, treated sample profile. So obviously it is having the advantages over the traditional 2D electrophoresis, which eliminate the gel to gel variation. This internal control provides the statistical confidence comparing the samples for the protein abundance changes. This is the most sensitive required less quantity of sample per gel. It reduces the number of gels needed to run the experiment. And as I have told you that number of gels requirement will be reduced if we are using the dyes compared to the 2D gels. So this was the gel-based approaches basically. Now coming to the non-gel based approaches. So most commonly used is the iTrack one. I'll just show in brief about that. This iTrack is the isobaric tag for relative and absolute quantification. So this is basically the isobaric tag. In this, we can go for multiplexing. There we have seen that 2 samples or 3 samples we can use it at a time. Here we can go for the 4 samples, 8 samples in a one experiment. Obviously, the statistical control with the duplicate and triplicate can be used. The identification and quantification simultaneously from the MSMS data. But in earlier methods, we use the gel-based approaches for the separation. Here in this method, we are separating on the liquid chromatography, which is the prior to the mass spectrometry. So this is called the, this is also the part of the LC-MS MSM. So separation is based on the liquid chromatography, and then workflow will be able to identify the biomarker, quantification, time course study, PTM, and then it is also having the simple workflow, reagent softwares, and the MS system is available, and then you can do it. So coming to how, what is this isobaric tag reagent basically? So this isobaric tag reagent is having the total mass of 145 Dalton, and mostly it is used on the peptide labeling basically. So when you label with this isobaric tag the peptide, it peptide mass by 145 Dalton. So this 145 Dalton is composed of the reporter ion as well as the valence ion and then the protein reactive group. So reporter ion is obviously for the reporting purpose, which will give the what is the expression level of that particular peptide, how many peptide molecules are there, which will tell you the further quantification. Then the valence ion is actually neutral mass. After MSMS, it will lose. But when you go for the peptide identification, this will be the important one because it will give the total mass is the same for all the peptides which is linked. So here we can see that peptide reacting group goes and bind to the molecule peptide to this peptide reacting group, and then this will total mass will be this. So this tag differ in between the reporter ion mass. Like when reporter ion mass is 114, the valence ion mass is 31. So 114 plus 31 is 145. Like this 115, 30, then 116, then 29, 117, 29. So if you take the 4 types of different tags, which is having this uh, different reporter ions, and then you can use label these 4 different sample and combine together and then go for the mass spec. So how it basically happens? Now you are having this 4 tags, you link with the peptide, okay, and then then mix this sample together and run it on the nano LC, separate it, and then you are getting the one mass which is having this 1352, which is the peptide identity, which will be the plus with this isobaric 145 Dalton. So when you fragment this mass with the MSMS, then you will get the Y ions and B ions, which will tell you that what is the sequence of this peptide. When you go for this reporter tag, which is 114, 115, the expression amount of this reporter tag will tell us that how much is the coming from that sample A, sample B, sample C, and sample D, if we are using the 4 different samples. So the expression, if it is all the same, that means it is the equal expression level in all the samples. If this one is higher, then you can say that you can find out the fold changes and then tell that this is like a 2-fold higher as compared to the control sample, as compared to the different sample. Like this, you can identify as well as the quantify it. So here is the another example where we are able to actually identify the reporter ion, B ion, and Y ion, and then identify what is this peptide, what is the protein it is coming from, and then you can get the identification and quantification. Similarly, now 8-plex are also available where you can label these different tags with the 8 sample and then go for the labeling and then combine and then go for one experiment where sometimes you can see that what is the biological different sample, you can use it, or the reproduce. If you wanted to run the technical duplicate, you can run one experiment. So this is the advantage is that. So it works with all the peptides, any class of the peptides, like phosphopeptide also you can able to identify. And with this multiplexing is possible. So this has a lot of advantage. It is more powerful experiment for the absolute quantification. Post-translation modification changes time course study. Wide dynamic range for the proteomics application. Allow the signal amplification in the MSMS. So basically for the biological system, when you are looking for the multiple samples, get the reproducible data, these techniques is highly reproducible and giving you the more accurate data as compared to the other traditional things. The recent one, what it is coming is the label-free quantification. But label-free quantification, as I told you in my introductory slide, that it is still not taking that momentum because this still has to be, lot of things has to be improved on that. So basically this label-free quantification based on the liquid chromatography. When you are separating your protein based on the liquid chromatography, the proteins will be eluted from one time point to the another time point. So when peak start eluting, it takes the, in this example, it takes the 20 seconds to elute this whole peak. Okay, so taking that advantage, the in the label-free approaches, the more data points has been generated. Like the data has been, quantitative data has been generated here with the different time codes. Like 20 seconds now divided into the 20 fractions. Like now 20 data points are there, and then getting the peak value. And based on that, then you can see that what is the expression level of that particular peptide. Okay, so this is more kind of use it into the quantitation in terms of the specificity. You are looking for the specific peptide expression level. So to summarize all these approaches, that I have not shown you the SILAC approach, but this is the mostly used for the cell culture only, as it is mostly used it for the, when you use it for the growing cell. Then the ITAC TMT are the same, which is on the peptide level. Then the label-free approach, which is most on the independent data analysis. So all these approaches, technologies are the different, but one has to decide what is the project, what we are looking for, what is my biological question, what is the method, suitable approach or method which will give us the correct answer out of it. And then design the experimental plan as well as the use the method or approach based on my, and then you can identify the proteins which are involved in that particular system. So it is very, still this field is evolving. Many more things are coming every day. So we need to be learning those new things and try to implement in our system. So if you come to the summary of this whole thing, what are the different applications are there? So first thing is the protein mining. So we can catalog all the proteins present in the tissue, cell or organelles, and then get the whole proteome profile, and then you can identify what are the proteins present in that. So you can make a database so that further people can use it for the further identification, quantification, see the expression level like this. And the most commonly used is the differential expression profiling. It is used for the identification protein from a sample as a function of a particular stage. Like you need to see the different cells differentiation from stage 1 to stage 2, then the different stages, then control versus disease, then the control versus treatment or the stimulus or anything. Then two different things you can compare and see that what are the proteins which are getting altered, and then you can further filter it out whether it is directly response or indirect response, and then go for the further downstream processing of that. You can also identify the post-translation modification, which are the most important event in the biological system, like phosphorylation, glycosylation, and oxidation. So these are the some of the applications which we can use it. So before going to the second part of my talk, if anybody is having the questions on these things, you can ask me or put it into the chat box, or should we go for the second talk?
Hello.
Hello.
क्वेश्चन आन्सर चालू आहे. So माझा पेपर कुठे गेला तुझ्याकडे तो राहिला ना काय झालं
Amol,
ते घ्यायला जाऊ.
if you are not tired, we can continue.
Okay.
And at the end, we can have our say question answer session.
Okay, sir. We will go to the second part now, and then we'll have the question
Sure.
answer.
Sure, sure.
Sir,
So
Vilas,
second part of
please
my
mute
talk is
yourself.
basically the understanding of the biology and diseases using this advanced quantitative proteomics approach. So I have already covered the definitions, then what is the proteome, what is the proteomics. So I'll not go in depth. So coming to the last slide, where I have stopped it. So as I have already told you guys that there are different types of application, what you are looking for. So based on the major application is in the clinical proteomics. Basically. So what is what is the clinical proteomics? Clinical proteomics is mainly the comparative study of the proteome associated with the healthy and the disease state in the search of diagnostic, prognostic, monitoring, biomarker, or as a target for the development of new therapeutic approaches. It also helps to understand the disease or the disease pathophysiology or the understanding the biology. So as I am talking about the clinical proteomics, what can be the sample for that? So there are different types of sample different people are using for the clinical research, where I have classified to the animal sample as the human sample. If you are going for the human study, definitely if it is a you can use the human tissue, whether it is normal tissue or in case of cancer, tumor tissue. Then the body fluid, which is commonly used as the plasma, serum, urine, CSF, saliva. And then see this system, understand this. Some people use the human cell line to mimic this thing and then try to understand you rather than the actual sample, they use it in the cell lines. Or they can go for the animal models basically. So in the animal samples, you can again go for the tissues or the biofluids, or you can use the animal cell lines as well. So, but the important thing is that what are the criteria we can put to select what sample to be used for the clinical research. So I have simplified this one that so it is always based on what is your research question. So if you are interested to understand the disease condition, or you are interested to identify the diagnostic marker or prognostic marker or therapeutic target, then you need to see that which one is the helpful sample for that. Okay. So based on that, you can select which is the suitable samples. Obviously, when you select the sample, you need to, if you are comparing it, you need to collect the suitable controls also. The minimum requirement is like age, sex, ethnicity, match sample should be there. Then there should not be any other variability like transporting and other things, so that your comparison should not give the false positive results. So obviously, then you also need to see that your sample should be either non-invasive or the less invasive. Because if you are using the clinical sample, it should not be a problem for the patient to give the sample. Then the ethical issues are one of the important things. Whatever the sample you are taking, so ethically it should be approved, permitted from at the clinical site as well as the research site also. And then definitely the number of sample required, what kind of question you are having. Based on the power statistic, you can use it and then find it out what sample, how much number we should use it. We can use it for the planning your research for the clinical proteomics. So as I was talking in my introduction slides, that these are the two types of proteomics are there. One is the untargeted, like discovery based the proteomics. And then another one is the targeted one, which is like a kind of specific targeted proteome. In discovery, which we are identifying more number of samples, as I have already explained, what are the different techniques, approaches are there. So basically, there we are using less number of samples, and then you are able to identify more number of targets. So now you are getting the more candidates for the biomarker, like 1,000 candidates you can get it from the 10 sample you are using in this study. But further, you need to go to the second step where you need to see that how you can verify this 1,000 target, and then see that how many molecules get verified or the validated. So either you can use the mass spectrometry based MRM approaches, PRM approach, or you can use the Western blot, ELISA, or the RT-PCR to validate this target. So obviously, not all proteins you are able to identify in discovery phase will get validated. So this will filter down to approximate 100, but for that definitely you need to use more number of samples. Then you go for the further validation using the different approaches where you can go down to the successfully targeting 5 proteins or peptide where you are using more number of samples. So you have to see it that what kind of study you are planning, and according, obviously the cost of the study will be increasing from discovery level to the validation level. As your sample number is increasing, number of techniques you are using is increasing. So you have to plan such a way that it should be give you the ultimate what you are looking for. So there are different challenges in the clinical proteomics. The most important is the nature and the quality of samples. As I told you that if you are collecting the human sample or the animal samples, you need to see that how much you can take it from that, which is a non-invasive method. Or based on that, we can design the experimental plan. Definitely, clinical sample you cannot take it in the quantities, so that your limitation is that limited amount of sample you have to use it for the further downstream processing and then identify the molecule. Then the sample handling comes into the picture, then the transport, then the deposit into the biobank. So your sample quality should be same for all the samples. If there is a variation in your samples, obviously this will lead you to give the wrong or the false positive results. Okay. Then the another thing is the sample nature. It should be compatible with the proteomics techniques. If you are using, say, serum sample, if you are using the tissue sample, in terms of tissue sample, you can see that there are more proteins present in that. But serum sample, as compared to the tissue, there are less protein. But if you see the albumin and the globulin are the major chunks, which is the 70% there. If you go for same sample further processing, you will only end up with those abundant proteins. So sometimes you need to deplete these samples and then definitely your experimental design should take care of the statistical power of sample size, the sample variability. There should not be bias in your designing of the experiment. So you should take care of the basement equality, the randomization, and avoid the heterogeneity. In the sample heterogeneous, then definitely you will end up with the false positive results, which will give you the wrong data in the future. Then another thing is the complexity of the proteome. So as I was telling you that as compared to genome, proteome is more complex, and when it comes to the post-translation modification and other things, then it becomes more difficult to identify all the protein at a time. So definitely in this kind of thing, you need to go for the further fractionation with some kind of strategies you have to use. You fractionate the sample. Sometimes people are using the proteome level, like the cellular level, nuclear proteome, then the membrane protein separately, cellular protein separately, and then doing it. Or they are fractionating based on the certain properties, and then going for the individual identification, and then combine this data, and then identify more number of proteins and the peptide. So important thing is that how much depth you wanted to go, you need to plan your sample processing according to that. Then the important thing in this is the quality control within and across the platform. Definitely, whatever the platform you are using, you should have the checkpoints in each and every step, so that you can tell that yes, whatever I have got the data is having the quality data and it is matching to the quality parameters. And then you should have your own QC checks in the workflow, so that you could, if somebody asks the question on your data, you can tell that okay, these are the QC checkpoints which tells us that our data is accurate in this up to this step. So these are the some of the challenges which we go. As I have already explained you that what are the different workflows approaches are there. This is the combined exam slide, workflow slide is there, where the sample preparation is the one of the important thing. Then this whether you use gel-based approaches, non-gel approaches comes into the sample labeling and this, and then go for the mass spectrometry. Multi-top, triple top, Q-top, triple quadruple, Q-trap, IRIN, these are all the names of the mass spectrometer. So, and then once you get this data from different samples, then you use the Proteoinformatics tools to analyze this data and identify differentially expressed proteins. So, so my lab works all these approaches in for the different projects. So my lab works basically on the clinical proteomics, where my focus is understanding of the biology and diseases using the advanced proteomics based approaches. So here, validate and characterize the protein involved in the different disease pathogenesis. Then once you identify these proteins, you need to determine the utility of this protein in terms of diagnosis, prognosis, then disease monitoring or disease progression, disease recurrence, or find out the new molecular targets for the development of the therapeutics or understanding the disease pathogenesis. We are also interested to identify the post-translation modification, especially phosphorylation, and their role in the disease pathogenesis, as most of the things happen through these PTMs only. As this diagram shows that my lab works on the different types of diseases which can be broadly classified into the cancer proteomics and then the viral proteomics. In cancer proteomics, my lab works on the Nasopharyngeal Carcinoma, that is the NPC, which is more prevalent in the northeastern state. And in the viral proteomics, my lab works on the different diseases like rabies virus, chikungunya virus, dengue virus. So basically, this will help us to understanding the disease pathogenesis and then maybe helpful to get a better diagnosis or the treatment in future. So coming with the small presentation on the what we are able to identify using the rabies proteomics data. I'm happy to tell that this project is in the collaboration with the Bombay Veterinary College Microbiology Department with Dr. Maji Madam and as well as Dr. Rajesh Parande. So this project started with the discussion with the Bombay Veterinary College, and as my interest is mostly the zoonotic diseases, I was more interested to start this rabies proteomics thing. To just give the small introduction, rabies is the zoonotic disease. It can affect both human and animals. Neurological disorder caused due to the rabies virus falls under the Rhabdoviridae family. The disease is 100% fatal, but it is preventable. The early stage rabies is very difficult to detect. If you see the worldwide, there are 50,000 human deaths occurs every year, and out of that 60% cases are being reported in India. This is mainly transmitted by dogs in India. The lacune, if you see in the literature, this is poor understanding of disease pathogenesis, lacks the sensitive laboratory diagnostic method or marker, and then no therapeutic available yet. So most of time when I present talks about the rabies proteomics, people ask, oh, this is the old age disease. Why we need to work it now on this? So if you see this slide, there are a lot of reports has come up to show that rabies is still there and rabies is still challenge for us, as this is the serious distressing disease, and that this is the challenge to the public health. So in this project, we defined the objective to identify differentially expressed proteins in the rabbit cases and the control during the infection using this quantitative proteomics approaches, then verify and validate this data of the differentially expressed protein, and then further characterize some of these proteins to decipher their probable role in the rabies pathogenesis. So we use the different workflows for this. So we had planned to start with the many different types of sample, like plasma, tissue, saliva. But in this study, we have used the first with the brain tissue sample. So sample preparation is one of the important things. Then then the proteomics identification, then validation and validation, and then the characterization. So first thing, once we get the samples from the brain tissue from the rabies infected and the control dog, we have gone for the identification using the traditional method, which is the cellular staining, the fluorescence antibody test, L gene PCR and G gene PCR to confirm whether these are the confirmed rabies positive. So those who have come positive has only included in this study. And then the control also, we make sure that this is the completely negative. So in this, we have used the eye-track approach, which is the couple with the 4-plex. So in this, we use the tissues, brain tissue, which lies this tissue, get the protein extracted, and then we go for the digestion of these proteins with the trypsin digestion, and then label these samples with the different isobaric tag, like 114, 115, 116, 117. So like these 4 samples we have taken, we are labeled with the different, and then pull this sample together, then go for the fractionation, and then run it on the nano LC, and then each fraction which acquire the data on the multi-top, and then acquired this LC multi data using the whatever the database search, and then this is the summary what we got it from this data. After doing the multiple eye-track experiment, we combine these things, check the reproducibility, and we could see that we could detect around 477 proteins with the high significant level, which are associated with more than 2,800 peptides, as well as the 5,100 spectra. We use the parameter like unused score more than 1.3, which is for the seeing whether it is the significant protein or not. Then we use further this data for the further analysis. Then check the control versus the infected samples, and then we put it like more than 1.5 fold is the upregulated and 0.66 fold is the downregulated, and we also check the statistical across the sample which is giving the more p-value which is less than equal to 0.05. So when we compare this, we could see that there are out of 477 proteins, 19 proteins are differentially expressed. In this 19 proteins, 10 proteins are downregulated, while the 9 proteins are upregulated. These are the proteins with their expression level with the fold change. It is plotted here. These are the proteins which are downregulated, while these are the proteins which are the upregulated. Then again, we use another approach, which is the eye-track 8-plates experiment. But here, instead of using LC mass spectrometer, we used latest very sensitive instrument, which is the Orbitap MS, to get the more number of proteins in that. So as I have already explained that we lyse the proteins, then digested with the trypsin. But instead of going for the 4-plex isobaric tag, I have used the 8 different isobaric tag and then label with the 8 different samples. And then after these samples are pulled together and then go for the fractionation. And then we learn each fraction on this Orbitap MS and then acquire the data. So we use run the multiple experiments or ITAC experiment and then get the reproducible data and then combining research with the proteome discoveries out there, and then we got the summary which we have put it here. So in this, we see that 2,281 proteins were significantly identified. So I wish to bring you the point here is that ELC multi approach only able to give 477 proteins, which are most abundant, but here we are using the more sensitive instrument, which is giving the more number of proteins, which are the low abundant also. So as these 2,281 proteins identified, which was associated with the more than 20,000 distinct peptides and the more than 47,000 distinct spectra. Again, we have put the parameters to check the fold change, which is 1.5 more than 1.5 fold, which is upregulated, and then 0.66, which is showing is the less than 0.66 downregulated. When we analyze this data further, we could see that there are 140 proteins which are the differentially expressed. Okay, out of that, 79 are the upregulated, and then the 61 proteins are the downregulated. We put the further statistical analysis and the further more filters into that, and then we could see that there are 40 proteins which are differentially expressed, wherein 14 are upregulated as well as the 26 are downregulated. This is the graph where we are showing this upregulated 14 proteins and then downregulated 26 proteins. So now we have got a lot of proteins, but how we can see that whether our data is reliable or not? The one important thing is that we are using the disease sample, which is the rabies. So we are curious to see that whether we can identify the rabies virus protein in that. Then we happily see that there are out of 5 proteins, we could able to identify 4 proteins in our mass spec data. So which shows that whatever the sample which we have used is confirmed rabies positive also, and then we are falling in the correct path. Then another thing is that as we are looking for the proteins which is the relevant for the human as well as the animal. So we are also interested to see that whatever the differentially expressed proteins which we could identify, whether these has human homologue, and then we search the databases and then based on the alignment and this, then we could see that this protein has all these proteins have the human homologue, and more majority of them are the more than 90% identical. So if we go further with this protein, if we can develop something, it can be useful for more human as well as the animal diagnosis or the treatment. So after getting all these differentially expressed proteins, then we are interested to see that what functionally annotate these proteins. So there are different softwares available for the functional annotation. One thing is the geo annotation. So we subjected this all these differentially expressed protein for the geo annotation, and then we could see that these proteins, majority of these proteins are localized into the membrane part or the different cell or organelle part. When we look for the molecular function, we could see that majority of proteins are involved in the different types of binding, like ion binding, small molecule binding, protein binding, or some type of catalytic activities like transferase activities, hydrolysis activities like this. So it shows that these are the important proteins. When you look at the biological processes, most of the proteins are enriched into the biological regulation or the some kind of cellular processes. We also go for the ingenuity pathway analysis and see that what are the top diseases, disorders are enriched with these molecules. We could see that it is showing the neurological disease and the psychological disorder are the diseases which are enriched in these molecules. So it shows that as these are the one of the important thing for the rabies infection, like it is a neurological disease, the psychological thing is also involved in that. So whatever the molecules we could identify in this data are more relevant as compared to the disease. So it gives us the confidence on our data that we are falling in the right path. We can go further for the characterization of those molecules, see what is the role of those molecules. We also go for the top molecular function as well as the cellular function. We could see that majority are involved in the molecular transfer or cell-to-cell signaling and the interaction. We could see that these molecules are showing the network interact with each other and form a network of majority of these molecules which are associated with the neurological disease as well as the psychological disorder. This red one are the upregulated while the green one are the downregulated one. So here two molecules are the HLA beta as well as DDX3X. DDX3X is the dead box helicase protein which bind to the virus protein and changes into the intracellular location. It provides alteration in the RNA structure, alteration in the translation initiation. So what we are seeing it here is that these two molecules are interact with each other with the interferon beta. So this looks like an important molecule. One, this HLA-B is involved in the immunological parameters, where the DDX3 is more involved in the viral replication. So we are further characterizing those proteins. We also go for the validation using the RT-PCR and the Western blot. And here we can show the successful validation of the 5 proteins, which is DDX3S, Ncbi, which are the downregulated, while these 3 proteins are upregulated, which is the matching to our this data. This results has been published in the Journal of Proteins and Proteomics recently. So if anybody is interested, can have a look in this paper and then see it. So further, we are interested to go for the characterization of those significant molecules. We are using the cell culture study and trying to see that what different time point these proteins are getting expressed. And is there any quantitative difference into the early stage versus the later stage of the infection? And we are also interested to go for the serum sample, saliva sample, or the CSF sample, which is more relevant for the rabies infection, and correlate this data. Can we able to identify some kind of marker, and we can then see the check the expression level of those proteins across the infection. So this is the with this rabies proteomics data. The another project is the Nasopharyngeal Carcinoma. I'll not go in detail of this thing, but this project is based on the cancer basically, which is more prevalent in the northeastern state. This is the tumor of nasopharynx. Usually this is the squamous cell carcinoma, and this nasopharyngeal mucosa also contains the other structure like salivary gland, lymphoid tissue, gives rise to the. So basically, this is the involved in the many organ in that particular region. So this is the rare type of malignancy in case of the worldwide, but it is most common in the Southeast Asia and some part of the Africa and Antarctica in comparison to the Western world. It is most prevalent in northeastern state of the India. The prevalence rate is 1.82% among the all cancer in this region. This is the 8th most common cancer in Northeast. So as this is most common in that, I collaborated with the Northeastern state different hospital. There are 6 different sites in the Northeast where we collaborated and then collected the different types of sample like the tissues, cancer tissue sample as well as the serum and plasma sample. And then to start with this, we started with the plasma proteomics. We use the multiple approaches like the gel-based proteomics approach as well as the iTrack approach in this. Here is the summary of all these approaches, what we could do. In the gel-based approaches, we could identify total 394 proteins in the NPC proteome profile, whereas the 423 proteins in the control sample. And when we analyze it further, we could see that there are 23 protein spots which are differentially expressed in the NPC, out of which 16 are highly expressed in NPC, where the 7 are highly expressed in the control plasma sample. When we go these disease spots and then go for the mass spec identification, we see that there we could able to identify 15 proteins out of each, where the 10 proteins are highly expressed in NPC, while 5 proteins are highly expressed in the control sample. Similarly, we use the ITAC approach also, and then with the repetitive experiments and then comparison with the gel thing, we could see that 9 differentially expressed proteins are identified with this the statistical score, where the 8 highly expressed proteins are in NPC, one is the highly expressed in the control sample. When we compile this data and compare with each other, and then we could see that there are 3 proteins, which are the serpina-3 vitamin D binding protein, retinol 4 protein, which are more common in all these samples. And these are the more important proteins. Further, we validated this data with the Western blot, showing that these proteins are more expressed into the NPC as compared to the control cells. We also go for the geo annotation. Then we could see that these proteins are enriched into the located into the protein complex extracellular region or the intracellular. So most of the proteins, as we are using the serum plasma sample, these are the secretory proteins. So it is expected to get these results. So when we look for the molecular function, we could see that again these are the involved in the binding activity or the transporter activity. And then we go for the biological processes, we could see that these proteins are enriched in the different biological processes, and these are the more diversified basically. So we have further gone for the IP analysis. This shows that the endocrine system disorder is the topmost disease which are enriched, and then the cell death and survival is the topmost cellular function, which is matching to the cancer thing. So whatever the proteins which we are able to identify seems to be more relevant, which need to be further in-depth study to find out the characterize those proteins. So I am not showing the other tissue proteomics data and the other project data. So in this project, we will be further characterizing those proteins. We are also interested to see the stage-specific manner tissue sample, what different stages if we can identify the signature molecule and their involvement into the particular disease using this proteomics approach. Obviously, we need to verify this data with a more number of patient samples using the targeted proteomics approaches. So I would like to thank my collaborators. This first put into the red are the collaborators for the my babies project. These other blue are the collaborators from my NPC project, and these are the collaborators for the JEV as well as chikungunya and the dengue things. I would like to acknowledge my lab members. Without their work, I could not have present this data. And then acknowledge the funding agency, DBT, ILS, Director ILS, and ILS conducts various types of training programs, workshop, conferences on proteomics also. If anybody is interested, they can check with the website or write to me. We are also works under the umbrella of the Proteomics Society of India. So we also conduct some kind of conferences under the proteomics. So if anyone is interested in the proteomics, start the proteomics or do some kind of proteomics, they can contact us, or they can participate in these conferences. Or I'll suggest you can be a part of the Proteomics Society, and then you can get much more information about the proteomics. So thank you. So I am open for the questions. Yeah, these are the some pictures from the conference, proteomics conference, international conference, which we conducted in the 2017. And we also conducted the workshops, symposium in 2016, 17, and then 18 also. So if there are any questions, so I'm open for it.
So the topic is open for discussion. If anyone has questions, please put it in the question box so that it can be addressed to Dr. Suryawanshi. Participants, please. Dr. Vaidya? Dr. Ingole, are there any questions?
I am not seeing any question in the chat box yet.
Yeah, Dr. Ingole, yeah, just a minute, sir. Dr. Ingole? Dr. Vaidya?
Hello, madam. Vaskar here.
Yeah. Dr. Vaskar, any questions?
No,
there
we could
are?
not see any questions in the chat box, but yes, Amal,
Yeah.
I have some doubts related to this, or you can say the just a curiosity point of view. What are the challenges you see in the proteomics? What challenges you foresee in the proteomics studies and applications as well?
See, sir, I see two types of basic challenges in the proteomics research. One is related to the sample, and another one is related to the technology. Okay, if we come to the sample thing, the sampling is the most important thing. If you are comparing two different types of samples, if your collection time, for example, if you are collecting the sample after infection 5 days and another sample is collecting after infection of 10 days, and you say that this is come under the same group, the profile might not be the same. Okay, so in that case, sampling classification should be done properly, collection should be done properly, and sometimes the transport. If we are collecting sample, like I face the problem with the Northeast. My samples are mostly collected in the clinical center at Northeast, and then we are transporting from there to here. So basically, this once they collect the sample, the time frame, like in 4 hours, 6 hour time, you need to process this sample, collect the serum or the plasma, then store into the -20, and then while transporting from there to here, you need to make the maintain the cold chain so that sample won't get deteriorated. So this is the one type of challenge. Second challenge is this proteomics technology. These technologies are still evolving. There are many things that's coming up. Today, whatever you feel that, okay, this is the ultimate, which we can do it, we can quantitate the protein, identify. But tomorrow, technology improvement is happening, which we can go for the some other alternative things which will able to answer the same question, what biological question we are raising, with the different techniques or with the using that advanced proteomics approach. So with the time we have to evolve it and see it that. That's why I was telling in one of my slides is that see what is your biological question, you see it that, and according to that you decide which is the sample you can take, what time point you can take, and you can answer your biological question. And then you decide, okay, if this technique is having some limitation, but that is not affecting my question, we can still go for that and justify it and publish the data or give some more knowledge about that. But if we are not able to justify with that technique, and then if you use it, then you may land up with the problem when you publish this data or convince to the scientific people. So these are the few challenges I really foresee.
Yes, thank you, Amol. It was a very nice presentation as well, and it was very nice to hear you after a pretty long time. Yes, we have posed one question that in case of GCMSMS process, there is a problem. I face problem with the nitrogen evaporator. It is from Dr. Rajesh Kumar Sahu. What he has said, I think you could also read that same.
Okay. So in this majority time, we are not using the GCMSMS. For the proteomics, we mainly focus on the LCMSMS. So for the metabolomic study, you can use it for the GC-MSMS, but for the proteomics, mainly our focus is on the LC-MSMS. Dr. Rajesh, could you get your answer?
Again, Amol.
Hmm.
No. Dr. Rajesh Kumar again, he has typed no, sir, about timing in evaporator. I think it seems to be too technical, or maybe.
Okay, so in GC-MSMS, you have to see it that nitrogen evaporator, what you are using, what kind of problem. This is the technical issue. You may talk to this vendor who has supplied it this, and then you see that how you can use it, whatever the problem you are facing. Because right now I am not able to tell what may be the exact problem in that.
Okay, well, Amol, apart
Yeah.
from the zoonotic disease, what you are focusing on, like maybe chikungunya, dengue, and rabies, what other disease, maybe some of the emerging or re-emerging disease, what you can forecast, where the proteomics will be more applicable as far as the diagnosis and the further relevance is concerned.
Sir, this question, I will more put it into the mode like we need to have the more clinical as well as the basic research combined together. Like clinicians and the scientists has to come together and then work it out which are the more relevant diseases. In terms of the human diseases, now COVID has come into the picture. So we are using this proteomics approach for COVID. And then we have recently published, not published, we have put it in the archive, one paper based on we have one of our colleagues has generated the hamster model for the COVID. So we compare the lung tissue at day 4 infection as well as the day 14 infection, and then put on the proteome profile and then see what are the molecules involved in that. And then now we are showing is that with day 4, what infection was there. Now after day 14, how this molecule actually comes into the recovery stage. The expression level has altered during the infection. After the 14 days, it is coming towards the recovery stage, and the expression levels of those molecules is restoring back. So in these emerging diseases also, this kind of work, we can do it and then try to see that what pathophysiological changes are there into the particular organs and if you see that for COVID also, it was very challenging that to tell how actually pathogenesis is happening in the body. So if we can get those answers, understanding of the disease is the one thing. Then how we can identify as early as possible, that is another thing. And the third thing is that is there any marker which we can use it like a prognostic or the before disease, whether we are more prone to that kind of some kind of these kind of things can be used using the proteomics approaches. So as I have told, diseases like COVID or some of the in between 2 years back, we were seeing it that encephalopathy or the AES, acute that one particular disease where the brain inflammation was more in the childs. So people are trying to do many more things using that thing. And if you see in case of the veterinary sciences, what I feel is that those which are the diseases challenging, like zoonotic diseases, rabies, or some other diseases, viral diseases, still we don't have any treatment for the viral diseases. Can we able to identify the earlier diagnosis as well as can we find out some therapeutic target for those viral diseases will be the good for the veterinary sciences.
Okay. Thank you, Amol. It was very nice to interact with you.
Yeah, thank you very much, sir.
Over to Matthew, madam, now.
So, Dr. Suryawanshi, at the outset, I thank you for giving us such a enlightening and such a beautiful lecture about proteomics. As usual, I know you are an expert in it, so we have got a lot to learn from you still. And my student has definitely, Dr. Farande, has benefited from your association, all of us, in fact. And it was such a nice piece of work that today also I feel that, you know, how we could do it, even I don't know. But your expertise has definitely enhanced our work on rabies, and I am sure we will carry it forward from there. All the participants, I think, are very happy with your lecture because they could get insights into different techniques you have used, like LCMS and mass spectroscopy, etc. Although I have never seen those machines before, and I don't know whether I will get a chance to see it, but definitely they have been of use, and we hope that this technology remains for a long time, and it will for a long time to come. And in veterinary, it is going to be one of those tools with which we can definitely use, as you said, for prognosis. And in the population. So I think prediction of diseases has become the need of the hour, and experts like you are going to be in this circle for a long time to come, sir. So with this, I put you over to Dr. Zende, the PI of NAHEP project, and I think he will give his concluding remarks. Thank you so much, Dr. Suryawanshi, and I wish you all the best.
Thank you, madam. Thank you so much for conducting smoothly this case. So, madam,
Thank you,
all
madam.
the participants are
For conducting smoothly
hello.
negotiation.
Am I audible?
So,
Hello, Dr. Vaskar,
hello.
I'm audible. Hello,
Yes, yes,
Hello.
yes.
Dr. Vaskar.
madam, am I audible?
Yes, 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 you are audible, sir.
Yeah. Madam, now we have a small concluding section. We have our university authorities also here, Dr. Somkur sir and Dr. Kurkure sir, Director of Research. And we have just 5 minutes concluding session will be there. And now we'll start that concluding session.
Sure, sir.
Dr. Ingole will start and followed by myself and other dignitaries.
Thank you all. This was a very good 5 days training session on bioinformatics, and I hope all the participants must have enjoyed, as well as they got a lot of information regarding, though it was a basic, but which was a need of our for we as veterinarians and other parties. To give their comments, first of all, I request the PI of this project and the convener of this training program, Dr. R.J. Jende, to give his comments on this program.
Okay, thank you so much, Dr. Ingole. Today we come to the end of this 5 days hectic international e-training program on introduction to the bioinformatics. I on behalf of my team and our authorities of the university, I thank all the participants. Today we have with us for this concluding session, Honorable Dr. Somkur sir is a Dean and Director of Instruction, MAPSU, and Honorable Dr. N.V. Kurkure sir, Director of Research, MAPSU Nagpur. Honorable Vice Chancellor, he is not able to attend because he is in another meeting, and all my HSP team here in our university, and participants and my colleagues. So, although I am not here in 100% involved in this all topics, but in between this all topics of topics right from
सर
day
एको
to
होते
the
तुमचं
last
व्हॉइस
day.
सर एको होते
Hello,
तुमचं गळा
Hello.
एको होते सर
acha.
हाँ
So now it is audible, madam. Hello.
Yes, yes, yes. Go ahead.
Yeah. So definitely on this occasion, I would like to thank all the speakers. They have delivered very nice topics, and with their rich experience in the bioinformatics, really they have delivered very good information to our all PG and PG student and the other faculty members of our MAPSU. And our university authorities are also thinking about that. Although we don't have this bioinformatics center at our university, we definitely will go forward for establishing such a nice bioinformatics center so that our students can get benefited of this. I thank right from the Dr. Kumaraswamy sir, Professor Department of Animal Genetics and Breeding, Dr. Anshuman Dixit sir, Senior Scientist, then Dr. Mohammed sir, Senior State University USA, and Dr. Amal Suryawanshi today. He has delivered talk on the advanced quantitative techniques for understanding biology and diseases. So from my heart and from my all Ncbi team and MAPSU, I really thank everyone, each and everyone, all the participants and the speakers for participating in this and concluding this 5 days training program. Thank you so much. Over to Dr. Ngole, please. Hello, Sompur sir. Hello. Sompur sir, please.
Hello. Hello, Dr. Jenny. Hello. Yes, Dr. Jende.
Hello. Sir, you are audible. You can speak. Hello. Hello.
Dr. Jende, am I audible?
Sir, you are audible. You are audible, please.
Actually, I came in the Office of Director of Research.
Sir.
My cabin, actually, I was not able to join because of some technical problem.
Okay,
so,
sir. Okay.
Is it over now? Dr. Kurkuri is also with me.
Okay.
Research, he is also with me. We are both joining on the same PC.
Okay.
Right?
Okay, sir.
So I think Dr. Kurkuri will say something regarding this program.
Yes.
You want to have an inauguration?
Yes, sir. Yes, sir.
Yeah.
Please.
Okay. So good afternoon, all. The NAHEPcast team of MAPSU Nagpur, they have organized this international training on bioinformatics. And this was a 5-day training. And what I'm seeing now that there are some more than 60 or 80 participants, those who are registered and attending this training. And this training has, in 5 days, they have 5 speakers, one speaker each day having 2 hours marathon lecture. And the topics covered were like bioinformatics, shotgun sequencing, microbial metagenomics, and microbial community analysis. Like that, very topics were there. And Last lecture today of Dr. Suryawanshi is regarding the proteomics and how we can use these techniques in our field, particularly microbial analysis, and as the NIHEP class program is for the food safety, so probably the lectures were targeted toward the application of these techniques in the food safety. First day I witnessed the training. I attended lectures by Dr. Kumar Sami from Kanuvas. It was a wonderful lecture and the presentation was also very good, and I hope same or with the higher scale they were for the other speakers too. The participants must have gained knowledge by attending this 5 days training, and because of this COVID, probably we are having this in an online mode. It would have been far better if we could have gathered together and run these experiments, at least touch these experiments in personally by analyzing some data. That would have been very nice, but unfortunately we can't do this, so we will not talk much about it. But participants now have the idea that how good these technologies are and how best they can use in their field. So participants may contact the individual speakers or the trainers for who are there in this training. So they may contact and get the details and or hands-on from them. And Dr. Jende, who is PI, and his team may facilitate the trainers for solving their problems in this field from these trainers. So with this presentation, I thank the organizers for arranging very nice training, and I thank the trainers that they have joined this training, and with the hope that they will use this training in their future professional life and do the laurels. So thanks for calling me here in this inaugural as well as valedictory function for this training. Thank you.
Thank you, Kurkure sir. Now I request any of the participants to give their views regarding this training program or their comments, if anyone who wants to speak on this platform.
Dr. Indole, Somkur sir is going to speak right now.
Okay.
You invite Dr.
Yeah. Somkur sir. Hello.
Hello. Yeah. Director of Research, Mapsu Nagpur, PI of the project, co-PIs. Today's presenter, I would like to say, Amal Suryawanshi, then committee members are there, and participants. Actually, since 5 days, this program is going on proteomics and others, bioinformatics and others. Means so many. Just now I heard from Dr. Kurkure. This program was going on, and yes, this training program when it is completed, after completion of the training program, whatever knowledge has been gained by participants, it is to be utilized by the participants. Then and then only we can see that yes, there is an outcome of any training program, and I hope that sincere efforts of these participants will be there to utilize the knowledge which they have gained from this training program. So just I would like to say thanks for giving me an opportunity, and I would like to congratulate the organizing team, that is Dr. Jende and his team, for organizing such international training program of 5 days duration, and hope the benefit will get to the participants. Thanks once again and congratulations to all. Thank you.
Thank you, Somkur sir. We are very much thankful to our Director of Research, Kurkure sir, and Director of Instruction and Dean, Dr. Somkur sir, for their constant support for these training programs and for this project. So now I request if any of the participants would like to give their comments regarding this program. Any participants? Okay. So thinking no comments. So
Sir,
we'll move
I would
towards-
like to say a few words.
Yeah.
I have attended all the days and I have found it really a very
Please,
insightful and
please
stimulating.
introduce yourself. Hello.
Hello.
You introduce yourself first.
Yeah, I'm Dr. Meera Shankaran Kutti, visiting faculty at SNDT Women's University Juhu campus. I'm a biotechnologist by training, working on food microbiology and food safety. I found the training program on bioinformatics very, very insightful and very relevant. Most of the speakers were very lucid and could explain very complex concepts in a very nice manner. And I really enjoyed the 5 days I spent in the training program and hope to participate in more such training programs in future. Thank you very much, the organizers, for organizing such a beautifully arranged program. Thank you.
Hello. Thank you, Meera. Hello. Any other participant?
Hello. Should I? Hello. Dr.
Yeah.
Yes, sir. Okay. So my name is Dr. Janish. I'm assistant professor in School of Public Health and Zoonosis, Gadwasu, Ludhiana, Punjab. So I really appreciate the efforts put by all the organizer to organizing this training course. It was really informative. We learned a lot regarding the bioinformatics, the different platforms we can use, the different softwares online as well as the others like R we can use for doing such kind of analysis, and really like the presentations by all the speakers like Dr. Amal, Dr. Maurice, Dr. Mitra, and Dr. Bhushan. The day he chaired the sessions, it was really interesting. So I really thank all the organizing team as well as all the speakers. Thank you very much. Thank you.
Thank you, Dr. Rashish. Anyone else? Okay. Thanks a lot for the participants. Yeah.
Yes, sir. I also have the same comments as the previous participants already remarked. But yes, it was really very enjoyable. And all these 5 days, I attended all the lectures. And I especially thank to Dr. Jhinde sir, to you, to Dr. Bhaskar sir, and the lectures which were particularly from Dr. Morris and Dr. Meera. They were really very insightful because they really also explained practically how to program R and other stuff. So yes, thank you so much, and please keep organizing such events. And
Yeah, Rashmi, can you introduce yourself? Can you introduce yourself?
Yes, myself, Rashmi Thakre Suryawanshi, and I was working as assistant professor at College of Food Technology, Udgir, but currently I'm a housewife right now, but I like to attend such programs, so I just registered.
Okay. Thanks, Rashmi. Okay. So I think we are coming to the fag end of this program, and I request Dr. Vaidya for vote of thanks.
Yeah. Thank you, sir. Am I audible, sir?
Yes.
Yes. Yes.
Yeah. So on behalf of Naheb Caste, Mapsu, Nagpur, and Mumbai Veterinary College, Mumbai, I extend a very thanks to all the dignitaries, speakers, different sessions, staff, and colleagues, students, and participants from India and other countries for joining us here. Of one international training program that is on introduction to microbiolomics. It was scheduled from 2021, organized under World Bank funded as a project. I would like to express sir, National Director, DDG Education, Sahib and Dr. Prasad Kumar sir, National Coordinator, for their support, guidance, always. Then I would like to thank our Honorable Vice Chancellor, Dr. M. Patrika sir, for their support and guidance. I also express my heartfelt thanks to Dr. A.P. Somkur sir, Director of Instruction and Field, MAPSU, for his guidance and providing encouragement to us. Thanks a lot, sir. Very much grateful to respected Dr. N.V. Kurkure sir, Director of Research, MAPSU, for their always guidance, support, and encouragement. Thank you very much, sir. We are grateful to Dr. V.D. Aher sir, Director of Extension Education, and Dr. P.G. Vasnik sir, Dean Faculty of Dairy Technology, MAPSU, for their constant inspiration, support, and encouragement. I express my heartfelt thanks to the speakers, Dr. Kumar Swami, Dr. Dixit, Dr. Maurice, Dr. Meera, and today's speaker, Dr. Amal Suryawanshi sir, for accepting our invitation in a very short period of time. Then I would like to thank our technical session committee chairman, respected Dr. N.V. Kurkure sir, respected Dr. Bhushan Jairao sir, Dr. S.D. Maji madam, then co-chairman, Dr. V.S. Vaskar sir, then reporter, Dr. M.P. Sawane sir, for their time and the session. I would like to thank our beloved Associate Dean and Course Director, Dr. A.S. Ranade sir, for his constant support, help, and encouragement for organizing this event. I also extend my heartfelt thanks to the Course Director and sir, then organizing secretary Dr. S.D. Ingole sir, Dr. M.P. Sawane sir, co-organizing secretaries Dr. R.V. Gaikwad sir, Dr. Mrs. R.S. Gandagi madam, Dr. and all OPIs who has taken a lot of efforts for organizing this wonderful event. Thanks to the program coordinator, Dr. Netra, Dr. Then we are happy to know participants. I also extend my thanks and welcome thanks to all the participants, those who were attended the different sessions. Because of them only this program was successful. And last but not the least, I appreciate the efforts taken by the team at NAEP and all the staff members and students of MAPSU. Thank you one and all. Thank you very much.
Thank you, Dr. Vaidya. So now I declare this program over. Thanks to all. Thanks to the participants.